So without further ado, I'd like to introduce today's speaker. This is Lorna McKellar from Bangor University, and her talk is entitled Where is Good? Evaluating and Applying Methods for Estimating Ecological Thresholds for Good Status. Thank you very much, Lorna.
Thank you. Yeah, hi everyone. Thanks for joining. So yeah, I'm Lorna. I'm based at Bangor University. I'm in my fourth and final year of my PhD. I'm part of the summer CDT, which is the Sustainable Management of Marine Resources. And as kind of part of the CDT, we're partnered up with different organisations as well. So I have Jan Hiddings, my kind of primary supervisor at Bangor University. I also have Christina Habon at JNCC, and then other supervisors as well, Abigail, of course, scholar at Plymouth, Tom Chagner at Exeter, and Seb Belanco at ICES, and Sally Kaiser and Ian Goring-Phillips at NIRAS as well. So yeah, the project is titled Setting Thresholds for Good Status in Marine Ecosystem Management. I thought, first of all, kind of useful to go through exactly how I'm defining what thresholds are in the context of marine ecosystem management for those that aren't aware. So we have different legislations in place at the moment in the UK and the EU, which are focused on assessing and then either kind of maintaining or improving the condition of our marine environments. So we've got the Marine Strategy Framework Directive in the EU and the UK Marine Strategy, which have this objective around achieving good environmental status in marine areas. The Area Beyond National Jurisdiction Agreement has objectives around maintaining and restoring ecosystem integrity. And then the EU Nature Restoration Regulation, again, has this objective around improving habitats from poor to a good condition, and actually defining that point of what is good environmental status, what is good condition, is quite difficult. And actually understanding when we've reached that point is when thresholds can come into place. Thresholds for good status define this boundary between when something is good and when something is degraded. So you can use them to assess, basically based on these kind of ecosystem state indicators, whether it sits above this threshold or below this threshold, you can assess what condition that area is in. So some of the challenges when actually estimating these thresholds for good status is, I included this figure here from one of Jan's papers, because I really think it kind of really nicely shows why this is so difficult. Because it's quite clear when you have these, super green bits when it's definitely really good, and these super red bits when it's definitely really bad. But it's that kind of muddy bit in the middle of, well, where do we actually kind of draw that line between where something is good and degraded? And at what point do we do that for kind of different indicators and regions can be quite challenging. So as a result, there's lots of different methods that are being used to estimate these thresholds for good status to kind of draw that line. Both quantitative and qualitative. And this kind of results in one sense, kind of a range of different thresholds, but in another sense, also kind of a lack of thresholds for some areas and some systems. So the kind of scope of this project was really to try and understand which methods should we be using and when should we be using them. So I'm going to today talk you through, as I said, I'm in my final year now. So I'm going to talk you through three kind of chapters that I've done, starting off with evaluating different quantitative methods, kind of taking this forward and applying some of those methods to data to actually estimate thresholds, and then also looking at a qualitative expert elicitation approach as well, and ultimately kind of informing more of a framework around kind of which methods perhaps we should be using with which kind of data, and try to kind of fill some of those gaps under where we are kind of missing thresholds under this marine legislation. So first of all, I'm going to talk you through the first kind of chapter which I looked at, which was evaluating different methods for estimating thresholds for good status. So in the kind of literature review, what I started doing was kind of looking at collating different methods and looking at how statistically robust they were, how kind of ecologically meaningful the thresholds they were actually defining were, and whether they kind of incorporated other social and economic aspects that kind of need to be included when we're thinking about kind of thresholds for marine areas where lots of other things are going on. And there are some reviews, especially into kind of how ecologically meaningful these methods are and whether they incorporate social and economic aspects. But particularly what I was not finding much information on was how statistically robust these different quantitative methods were and how that would kind of inform which method we perhaps should be using for certain types of data. So that's what I focused on with this chapter, was trying to evaluate how statistically robust different methods for estimating thresholds for good status were. So we wanted to answer some questions around how these methods would respond to different types of pressure-state relationship, different levels of noise in the kind of indicator data, and different sample sizes as well. So how would that actually impact the thresholds that they're estimating? So I looked at 4 different methods. 2 methods use reference condition data, and then I'll talk through the other 2 methods afterwards. So here we've got range of natural variation and statistically detectable change. So range of natural variation uses, or they both use reference condition data, which is how we're defining it, is data from indicators that are within kind of undisturbed areas or least disturbed or minimally impacted areas. So you kind of monitor it over time. And then what range of natural variation looks to do is quantify that full range of natural variation and then estimate the threshold by just kind of taking a lower percentile of that full range just to account for any outliers. You would then apply this threshold to areas that are experiencing pressure to assess whether they're in a good state or not, whether it falls above or below the threshold. Statistically detectable change is another method which uses the same data, but instead of kind of that full range, it just uses confidence intervals, or the way we applied it just uses confidence intervals. So it takes that lower 2.5 confidence interval and uses that as the threshold to apply to areas that are experiencing pressure. So statistically detectable change is slightly less ecologically meaningful because it's more about the kind of power of that test and that confidence interval when you're thinking about range in comparison to range of natural variation, which is really looking at how that system varies naturally in its kind of undisturbed state and just takes the kind of almost the full range there. The other 2 methods we looked at were use pressure state data. So this is tipping points in terms of kind of ecological tipping points, and then the other method, distance to degradation. So pressure state data, what I mean by that is how the ecosystem state varies as a function of increasing pressure and what that relationship between those 2 variables looks like. So for the tipping points method, you would want to see a kind of concave tolerant relationship. So there's a point at which just after the ecosystem state kind of disproportionately declines with just a small increase in pressure. And so you'd want to estimate your threshold just before that point by identifying the kind of this critical angle on the slope on that curve. And then, yeah, estimating your threshold there. So anything above that would be in a good state and below a degraded state. Distance degradation is a different method, which kind of uses a very, at first, the same approach as tipping points in terms of identifying this kind of critical slope angle on that curve. But because you're looking here at a convex relationship, when you identify that point, it's already, the authors defined it as the degradation point because it's already at the point at which the system is mostly degraded. So in order to estimate a threshold for good status, in order to kind of push that further up the curve, what they suggest doing is essentially adding a percentile onto that degradation point, which is a percentile of the range between the kind of maximum state value and that degradation point. So it's, again, slightly less ecologically meaningful when we think about tipping points, because even though that percentile the authors define based on the kind of sensitivity of the system, it's still ultimately an arbitrary value that we're adding on top of that degradation point to define the threshold there. So those are the 4 methods. And then what we wanted to do, because we want to see how these methods estimate thresholds based on kind of different types of data. We simulated indicator data. We simulated 3 pressure-state relationships, so linear, convex or sensitive, and then tolerant concave, and then the reference condition data as well for those first 2 methods I mentioned. We varied the level of noise, so 6 levels of kind of added noise, and then also the sample size as well, so 5 different sample sizes. And that's an example of one of the sample sizes across kind of different levels of noise. So each row is a different pressure state relationship. You can see those curves quite nicely on the left-hand side. And then at the bottom is that reference condition data. So just a kind of time series of monitoring data there. And then as you move from left to right, you can see the different levels of added noise, and those shapes and those curves start to disappear as the noise increases. So we fed that data into those 4 methods. So obviously the pressure state data for tipping points and distance degradation, and the reference condition data for range of natural variation and statistically detectable change. And this is just an example of the first kind of simulated data I showed you a second ago, with the thresholds plotted on top that these methods estimated. So the thresholds are those dashed lines. So distance degradation in purple and tipping points in blue. And then statistically detectable change in red and range of natural variation in green down the bottom. So if I start at the top, you can see that distance degradation has estimated thresholds for these first 3 levels of noise. But then as you continue on, there's no kind of curve that's been fitted and no threshold that's been estimated there. A similar kind of story for the tolerant concave relationships. So again, distance degradation and tipping points estimating thresholds for those first 2 data sets, but we're missing them for the rest of those levels of noise. There's no threshold estimated for the linear relationships because there's no kind of change in slope on that relationship there. And then reference conditions methods down the bottom, you can see that there is a threshold estimated from statistically detectable change and range of natural variation for every single one of those different levels of noise. But obviously, as the noise increases, the range of natural variation threshold gets much, much lower because the full range of natural variation is increasing so much. And it's assuming that all of that variation in there is natural variation rather than kind of added noise. So what we did was run this 100 times for each of those different data sets and then collate that together, which is what this figure is showing here. Again, just looking at the added noise. So this is just a single sample size. So you can see a similar kind of story as I just went through. Statistically detectable change and range of natural variation, those top 2 plots there in red. As the level of noise increases, the thresholds estimated by range of natural variation in particular get much, much lower. Not so much of an effect with statistically detectable change. And then distance degradation and tipping points, those bottom 2 plots. You can see here that across the kind of board, the average threshold that they're both estimating is very similar, all the way up to 90% added noise. But you have these numbers on these 2 plots here, which essentially are the proportion of thresholds that have been estimated out of 100 runs. So 1 would be, there's a threshold estimated for every single one of those data sets. But then as you move from left to right with that added noise, you can see that the proportion of thresholds that they're estimating kind of declines quite dramatically. Even when you're getting up to kind of 50% added noise, there's only kind of, for distance degradation, either 20% or 50% of the time a threshold is being estimated. And for tipping points, around 20% of the time. So there's quite a low rate of estimation as the added noise increases. And then this is looking at the different levels of the different sample sizes on the x-axis now. So again, starting at the top, statistically detectable change and range of natural variation. I should also say there's no numbers on these ones because they're estimating thresholds for every single data set. So the sample size really impacts the statistically detectable change thresholds much more than range of natural variation because it's using confidence intervals. The smaller the sample size, the wider those confidence intervals get, whereas that's not so much of doesn't have as much of an effect on range of natural variation. Again, a similar kind of story for distance degradation and tipping points. The average threshold kind of across these different sample sizes is quite similar. but the actual proportion of thresholds that's being estimated is really quite low. This is at 50% level of added noise. And then also at the smaller sample sizes, the variation in thresholds is getting quite large as well. And they're estimating thresholds above 1, which is kind of theoretically impossible as well. So because the thresholds are a relative value between 0 and 1 or 100%, so can't really go above 100% of the reference condition value. So what we kind of summarized from that is that, yes, the methods using this pressure state data, so chicken point statistical degradation, are kind of more accurate across these different levels of noise and different sample sizes. There's much lower reliability in whether they're actually going to estimate a threshold or perhaps estimate quite a spurious threshold if there's a high level of noise or a low sample size. Whereas these methods using reference conditions, rate of natural variation, and statistical change are more reliable in that they're always going to estimate a threshold. But it's important to be aware of the fact that for each of these methods, either greater levels of noise or smaller sample size data is going to impact where that threshold ultimately gets estimated as well. But overall, these methods using reference conditions are more reliable and a bit more transparent in how they're actually estimating these thresholds, especially if data availability is limited as well. So that brings us on to the second chapter, which was essentially taking some of these methods that we evaluated and applying them to empirical data sets to actually estimate thresholds. So we needed to find reference condition data. So as I said, these kind of minimally impacted time series. So we wanted to find this kind of long-term monitoring, as I said, of minimally impacted systems to kind of try and look at what the range of natural variation would be around a mean in a system that was experiencing a minimal amount of pressure and therefore good. So range of natural variation, as I said, uses a percentile of that full range of natural variation, so RNV, and then statistically detectable change, SDC, uses a confidence interval, so the lower confidence interval there. So the data we found, so we were looking for just biomass and abundance data, essentially, and minimally impacted. And we found this for 4 different systems. So we had plankton abundance data across 3 life forms from the Continuous Plankton Recorder Survey from 1960 to 1980. We had shoot density data from seagrass beds in the Isles of Scilly and Scoma Marine Conservation Zone, which was minimally impacted through expert kind of knowledge of those that are collecting the data and working within those systems. And then we had tropical reef fish biomass across 4 broad trophic groups from NOAA, from 10 uninhabited Pacific islands. And finally, we had benthic invertebrate biomass data for 33 kind of single species monitoring time series, and then 31 total community kind of monitoring time series as well. And that was through a lot of work done by one of Jan's previous master's students, Abigail Nichols, and through data repositories such as Merman, through researchers at Bangor, through Natural Resource Wales as well. And that was deemed minimal, to have minimal physical disturbance. We looked at the sites through Global Fishing Watch. We asked the experts who were involved in collecting the data, and we looked at the papers that in which the data was published as well to see if there were any other pressures mentioned in those areas. So I think it was about just over 90 data sets in total. And so we wanted to estimate thresholds for every single one of those data sets. So I just wanted to show an example of how we did that and how we kind of standardized the data. So this is a one data set from a single species benthic invertebrate biomass data set, blue crab density from 1990 to 2020. And then what we do is we standardize the data so that the mean of that data is 1. And we're looking at the variation around this mean. And essentially looking at the variation underneath 1, so from 0 to 1, that variation underneath there, which ultimately produces a relative threshold. So it's a proportion of that reference condition mean, essentially. So range of natural variation threshold here would be 0.77, so 77% of that reference condition value. And then statistically detectable change would be 0.87. And so always the thresholds are ranging from 0 to 1, and 1 would be your kind of assumed carrying capacity of that system. So we applied those two methods to all of those data sets and then collated them here based on the different kind of species groups. So if I start on the left, we've got benthic invertebrate biomass, total communities, and then single species, plankton life form abundance for large copepods, meroplankton and small copepods, and tropical reef fish for biomass for trophic, four broad trophic groups, piscivore, planktivore, primary and secondary, and then shoot density data for seagrass. Range of natural variation thresholds are in red and statistically detectable change thresholds are in blue. So range of natural variation is that kind of percentile of that full range, and then statistically detectable change is that kind of confidence interval threshold. So you can see if I start on the left, there's quite a bit of variation in the thresholds here, especially for those kind of community data sets, ranging all the way from 0.2 up to 0.9. So quite a bit of variation there. So the plankton threshold, plankton abundance threshold, you can see there's quite a bit of disparity between those estimated using range of natural variation and statistically detectable change, because it's quite a long time series, but it has a lot of variation in it. Ultimately, the range of natural variation thresholds are much lower than statistically detectable change. Whereas for the tropical reef fish trophy group threshold, you can see not as much of a disparity, but the range of natural variation thresholds are higher, but statistically detectable change, because the time series are much shorter. That really impacts the threshold that statistically detectable change estimates with those confidence intervals. And then finally, we have the seagrass shoot density thresholds, which we only had 6 data sets, but there's really not a lot of variation in there at all. Most of them were from the Isles of Scilly. We had one from the Scoma Marine Conservation Zone, but yeah, really ranging from maybe 0.7 to 0.8 for range of natural variation, and then maybe kind of 0.8 to 0.9 for statistically detectable change, so much less variation there. And then you can see the mean thresholds for each of those groups, those filled in circles there. But what we wanted to do kind of beyond providing kind of a mean binary threshold for each of these systems is to try to model some of that variation to look at the uncertainty in the thresholds. So we went ahead and looked at the probability density distributions of each of those systems. I'm just showing you the benthic invertebrate species and communities just for clarity's sake, so we can zoom in on it a bit more. But what we did here was kind of, as I said, model that probability density distribution based on that variation that you just saw, to look at, well, what's the probability of a system being in good status, rather than just kind of a binary yes or no threshold value. So what you could do here is, if we look at the benthic invertebrate communities, if you looked at a threshold of 0.57, there'd be a 50% probability that that system, that region was in a good state. Whereas if you looked at a threshold of 0.82, you could say there would be a 90% probability that that system is in a good state. And I've included that in a table here on the right-hand side, but that kind of gives a bit more information around kind of how certain we are whether or not an area is in good environmental status or good status even, and gives decision makers a bit more of a role in deciding what is an appropriate probability of being in good status as well. The other thing we did with this study was look at whether we could account for some of the variation in those thresholds that you just saw. So again, we did this for all systems, but I'm just showing you the benthic invertebrate species, again, just for clarity. So for this system, we had 33 thresholds from 33 data sets, and we looked at the depth of each monitoring site, the sediment type, and the maximum average lifespan of the species. And we found that the depth and the maximum average lifespan significantly predicted the range of natural variation threshold, with those that had greater maximum average lifespans and at deeper depth having higher thresholds, suggesting that there was less variation in their biomass over time. And what this kind of potentially allows us to do as well is start to think about predicting thresholds for areas or species with these specific life history traits or specific environmental characteristics. So for a particular depth or for a species with a particular maximum average lifespan, we can predict what the threshold would be, perhaps if we don't have the data available to do it, as I've done it so far. So yeah, just to summarize this chapter, what we've done with these methods is shown that we can actually estimate the probability of a system being in good status, which, as I said, useful for decision makers and moves us beyond the kind of binary threshold values. We've also shown that you can potentially predict thresholds for areas using these kind of general environmental characteristic and species life history relationships, which can be particularly useful where there's a lack of data availability. And then finally, what we've done is kind of set out this framework in a way that can be applied to indicators that we haven't included or data that we haven't included in order to estimate kind of transparent, consistent thresholds and potentially fill some of those gaps in assessments where they're missing at the moment. So, oh yeah, I just wanted to give a quick shout out to people that helped me gather data for this because it took a really long time. And yeah, I really appreciate yeah, them sharing the data that they did as well. So big thank you to everyone on the screen. And then my final thing I wanted to talk through today was the qualitative side of things, which was actually asking experts to estimate thresholds. So I've shown you this figure already. I think it's nice just to remind ourselves that there is a challenge in actually defining what good is for a lot of systems and defining where, at what point do we kind of draw that boundary. And this is done with quantitative methods, which I've spoken through, which are kind of, you know, range of aspirations, distance detectable change, distance degradation, tipping points. They're very consistent in most of the time in the way they're applied, which makes them kind of also much less, they're much less subjective and usually much more transparent than some kind of qualitative expert elicitation approaches. But they're also most of the time quite dependent on data and the way, like conversely, the way in which they can be applied is much less flexible because they're so kind of consistent and transparent and less subjective. On the reverse side of things, the qualitative methods, expert elicitation, they don't require as much data most of the time, and they're much more flexible in the way that they can be applied and the way you can kind of incorporate other things into how we're estimating the thresholds, such as the kind of social and economic aspects. But on the other hand, they're also much more subjective and potentially subject to biases as well, especially when they're used in a much more unstructured way. So this is what we wanted to kind of address with a structured expert elicitation approach to actually estimate thresholds. And we used the IDEA protocol by Victoria Hemming and colleagues, which stands for investigate, discuss, evaluate, aggregate. And essentially it obtains expert judgment in a much more kind of formal, controlled, systematic way in order to reduce some of these biases that are kind of inherently in most of our human judgment and decision-making processes, such as some of those kind of group biases, groupthink, the halo effect, also those individual biases such as anchoring and adjustment and availability, and then overconfidence in estimates as well. So it's kind of a modified, they describe it as a modified Delphi approach. So you start off with a survey and a kind of a private response survey that people complete individually. We wanted to focus on estimating thresholds for seafloor integrity, which is the descriptor 6 of the Marine Strategy Framework Directive, and I believe the UK Marine Strategy. And it's around the condition of our benthic systems. So we had 6 questions in our survey. We wanted to look at 3 different benthic kind of example systems. We wanted to use the same group of experts for all these systems. So we wanted them to be similar enough that we could ask the same group of experts, but also potentially a little bit different in their characteristics that we might elicit different thresholds for different systems. So we looked at a modiolus bed, a horse mussel bed, a soft sediment bivalve community, and a common cockle bed. And under the Marine Strategy Framework Directive and UK Marine Strategy, we need both a quality threshold, which is looking at the condition of our marine environment or these regions, these systems, but also the extent of this condition. So what proportion of that area is meeting that quality threshold for that overall area to achieve good status. So this is just a little bit of an example of the kind of first question. So in the survey, we gave as much information without kind of overwhelming people about these different systems, about specific case study areas, about information about the kind of functioning of those different systems, and presented it as kind of a literature review, a mini literature review of these different systems, but invited experts to, use any other expertise or knowledge or kind of look up any other information they wanted to. So these are two of the questions we used for the modulus bed. So the first one was the quality threshold. So assume that a density of a modulus bed in pristine condition is 600 per meter square. What density of mussels per meter square needs to remain to avoid adverse effects on the structure and function of this bed? The second question, very similar, but thinking about the extent of a modulus bed, what percentage of the area needs to remain to avoid adverse effects on the structure and function of this bed? So first a quality threshold, then an extent threshold. And within those kind of 6 overarching questions, we used a 4-step elicitation procedure, which the authors of the IDEA protocol recommend. And that asks experts for a lowest plausible value, highest plausible value, and most likely estimate, and then also ask them, how confident are you that your interval from lowest to highest actually captures the true value? And then asks them to enter a number between 50 and 100 on the basis that if they were to enter a number lower than 50, it essentially says that they're more confident that the true value is outside of their interval. So we basically ask them to make that interval wider in order to get at least 50% confidence above that their interval has the kind of true value inside. So we asked experts to complete the survey and send it back to us. And then we got them together in different group discussions based on kind of availability, in which they were able to view other experts' anonymous estimates, and then also discuss the kind of reasoning behind their estimates, the approach they took, how they interpreted things like adversely affected structure, functioning, in order to kind of share knowledge, share kind of expertise, and yeah, just have a bit more of a considered judgment before we ask them to go back through their survey and potentially change estimates off the back of kind of having that conversation with other experts. Important to note that we didn't ask them to agree on a single value because there is evidence kind of suggesting that that doesn't necessarily equate to greater certainty in a value just because you have a consensus, potentially more linked to kind of conformity, linking back to that kind of halo effect bias, and then also potentially just expert fatigue and agreeing rather than actually kind of being more certain in that value. So we didn't ask them to agree, and we instead were going to look at trying to model the variation in estimates to inform, again, that kind of uncertainty value around the thresholds, rather than, again, providing that kind of binary single value. So we had 21 experts take part across, with expertise across kind of benthic ecology, assessment and indicators, and across academic, government and consultancy organisations, mostly UK-based, but across a few other EU countries as well. And so I'm going to show you the thresholds that they estimated grouped by the different questions. But just to kind of reiterate, the question that they were answering was what biomass or extent has to remain to avoid adverse effects on the structure and function of that system. So you have the, these are the thresholds. So each box plot is kind of 21 expert thresholds grouped together. And then you have the questions down the bottom. So horse mussel bed quality, extent, soft sediment bivalve community quality, extent, common copper bed quality, extent. And you've got the mean thresholds down the bottom there. And that's, again, a percentage of the kind of undisturbed state that has to remain to avoid adverse effects on the structure and function of that system. So you can see, so the modulus quality threshold was highest out of these 3 systems. It was deemed kind of most sensitive and most long-lived and also slower to recover. Then it was the common cocklebird, which was considered less sensitive, but also considered an important prey species for wading shorebirds. And then finally was the soft sediment bivalve community, which was considered less sensitive. And the data provided showed there was kind of quite high variability in biomass over time. So it was deemed kind of quite quick to recover as well. I think, interestingly, the extent thresholds that experts estimated were always higher than the corresponding quality threshold for all three of those systems. And that was kind of considered quite important, again, for recovery and, ensuring there was enough of a population to allow that system to kind of recover from maybe kind of lower quality thresholds. But as I said, there's quite a lot of variation, especially in some of these soft sediment bivalve community thresholds as well. So what we did was we had this group discussion, and we went through and did a thematic analysis of that group discussion. So kind of going through the qualitative statements from experts and kind of grouping together themes and seeing kind of what came out of that. And something that we found quite interesting was the way in which experts were interpreting what not adversely affected was. So the Marine Strategy Framework Directive defines it like this. This is what we gave experts. So particularly, just to kind of highlight some bits, it says that the ecosystem should maintain its respective components along with its natural levels of diversity, productivity, and dynamic ecological processes, so structure and functioning. It also says the levels of disturbance must be at a level that ensures the dynamic recovery potential is maintained. So we highlighted there was this first interpretation, which we called adverse function, which was more focused on the kind of current state of the system and whether and what kind of proportion of its state that it had lost before it would be considered not adversely affected. So it was more about that kind of functioning and what the current state of the system was in now and whether that was good or not. There was this kind of second interpretation we felt around, called recovery potential, around whether or not the system could recover to a kind of a good state or more of a kind of natural levels in the future if pressures were removed. So there was kind of, it felt like these two quite different interpretations here, but both quite clearly linked to that definition that we gave around what not adversely affected was, that's from the Marine Strategy Framework Directive. So what we did was go through each expert's threshold and code them based on whether we felt they fell into either of these two interpretations. And so that's what you can see here. So green is that adverse function, the functioning currently in a good state, and red, that recovery potential, could it recover to a good state in the future? So not so much for the modiolus quality and extent thresholds, but for the soft sediment bivalve community and the common cocklebirds thresholds, there was quite a clear kind of separation in those two interpretations. In the adverse function was much higher, and then recovery potential thresholds were much lower for systems that were deemed to be able to recover quite quickly. Even though we've now kind of highlighted some differences between these thresholds, there's still quite a lot of variation in these thresholds, ranging from, kind of for the modiolus quality from kind of 20 to 90%. So as we said, what we were going to do is model some of this variation to look at the uncertainty around these thresholds. And that's what this is showing here. So for each of those questions, we've modeled each of those threshold distributions based on, again, those two interpretations of what not adversely affected is. So in a similar fashion, as I said earlier, what you can do with this is start to estimate what the probability of a system being in a good state is based on particular threshold values. So for example, the horse muscle quality thresholds, both interpretations were quite similar, gave quite similar distributions. So let's say there was a horse muscle bed that had 60% of its kind of unimpacted reference state. According to these distributions, adverse function, there's a 45% probability it's currently in a good state. And then the recovery potential, there's a 50% probability that it could recover to a good state. Whereas if you looked at something like the common soft sediment bivalve community thresholds, there's quite a disparity between those two distributions. So let's say there was a bivalve community with 20% of its kind of unimpacted biomass. According to the adverse function distribution, there's only a 2% probability it's currently in a good state. whereas that recovery potential distribution suggests that there's a 50% probability that it could recover to a good state in the future. So we also wanted to compare this with those distributions that you saw earlier on, so those quantitative threshold distributions. So I've just got the benthic invertebrate community ones here, just to compare. And you can kind of see for most of these that these are quite a bit higher than the thresholds that were estimated, those quality thresholds that were estimated. It's similar to the horse muscle quality distribution, but overall, the kind of average quality threshold that experts estimated was 0.52, which is quite a bit, which is a bit lower than these benthic invertebrate thresholds estimated using those quantitative methods. So just to kind of summarize this, what we've shown is that the IDEA protocol can estimate thresholds very transparently in a kind of replicable approach with very limited data. We have estimated thresholds, average extent threshold of 74% and an average quality threshold of 52%. Yes, there is high variation in the thresholds that were estimated, but we can show that we can use this to model some of the uncertainty and look at what the probability of the system being in good status is. But there wasn't as much alignment with some of the quantitative approaches. The thresholds were a little bit lower. The expert estimated thresholds were a little bit lower. I think quite importantly from this study is the fact that using the term adversely affected and the MSFD definition is that this elicited two different interpretations of essentially what good was for these systems. So one thinking about the current state of the system and another thinking about whether that system could recover in the future. So I think we quite clearly need to think about, is it one or the other or both? And actually quite clearly defining these upfront is going to be really important in order to use this approach to kind of estimate a bit more maybe applicable thresholds to actually use in assessments. And yeah, so that's what we kind of summarized, was that expert elicitation would be especially useful for defining some of those key terms and supplementing some of these threshold setting approaches as well. So my last slide, just to kind of summarize those three chapters that I've spoken about. So the first one was looking at simulating thresholds to evaluate how statistically robust these different methods were. And we found that the reference condition methods were a bit more straightforward and reliable in estimating thresholds, especially with limited data. The second study we looked at was defining this framework for how to actually estimate thresholds using reference conditions and looking more at the probability of a system being in good state rather than those binary values. And then we also showed how this could be applied independently to other indicators to estimate ecologically meaningful and potentially much more scalable thresholds using those predictive relationships. And then finally, we applied a qualitative structural elicitation approach to estimate replicable, quite transparent thresholds and how we did it to account for these biases, but showed that defining those key terms upfront is really, really key for actually estimating applicable thresholds for assessments. Just to say a massive thank you to all of my supervisors who have been amazing throughout I think over 3 years now. So yeah, and thank you guys for listening as well.
Thank you so much, Lorna, for such an interesting talk. We've got a lot of questions and maybe not enough time, so I'm just going to dive straight in. So first question, did you account habitat data to be in recovery phases, which may possibly affect the results, as I believe areas where seagrass data were taken from restoration areas? I think that's relating to chapter 2.
Okay. From my understanding, they weren't, and correct me if I'm wrong, but I don't think they were, especially for the Isles of Scilly, they weren't so much linked to restoration. Only because the data they gave me was it was quite a long time series. But no, that's a really good point to think about. And I haven't looked at kind of restored systems, so seagrass and kind of other kind of bivalve systems, and I haven't found as much data on them. I think because most of them are under a lot of kind of restoration projects, I haven't found the kind of reference condition time series that I've been looking for. But yeah, it would be really interesting in looking at how some of those kind of restored time series and what kind of thresholds that we'd estimate there and how they would, I guess, compare to ones that haven't been restored as well. But yeah.
Thank you. Next question is, do you have views on avoiding shifting baselines using existing models?
Yes, that's a good question. So we haven't looked at shifting baselines. So for that second chapter where we kind of standardize the data, what I didn't say and what I should have said also is that we fit, if there is kind of a shifting baseline, we fit a linear model through that to only look at the variation around that kind of linear model rather than looking at that kind of full shifting baseline there. So no, I haven't looked at it, but yeah, I would be really interested in trying to explore that more in the future.
Were other traits included in the analysis, such as fragility or position on the sediment?
No, they weren't included. I think that's more of a data availability aspect on my behalf. And then, also, I think I would be interested in looking at other kind of life history traits and environmental factors, but then also I think not so much of an issue for the benthic stuff, but also for other systems not wanting, I didn't have loads of data sets and not wanting to include kind of too many variables as well there. But no, I didn't look at that, but I would be interested if we had that information.
Did you take into account the effects of confounding variables, particularly those from environmental factors?
Well, it's a broad question. Is that something related to something specific?
That's the only
factors of... Okay.
It doesn't help that we have a bit of a word limit as well with the questions.
Okay. Confounding factors. I'm sorry, I don't really know how to answer that without more information as to what it's in relation to.
Unless maybe the person who submitted the question wants to jump in, perhaps, or maybe put their hand up later. We can always come back to that.
Yeah, sorry.
That's all right. Might you include further data sets in the future, and what is the quality coverage of your data sets like?
I think at the moment the PhD is coming to an end, so there's not as much scope to look at it in the future. I would be interested in kind of carrying on the work. I'm at the moment I'm involved in a kind of advice request with ICES, which is had a data call, not just specifically for these reference conditions, other data sets as well and other methods, but I will be applying those similar methods to these. Obviously that's kind of more specifically benthic related, but yeah, I will be kind of expanding on some of that in that request there. But yeah, maybe a postdoc afterwards.
What are realistic timescales for improving ecosystems and getting them reassessed?
That's a good question. I think in terms of reassessing systems, I think that would be more down to the kind of legislation maybe, and the kind of timeline of they have for kind of assessing or reevaluating every 6 years or every 3 years. But that's a really good question. Again, I haven't looked so much at the restoration aspects of this, but yeah, it would be interesting to see how they compare to those different thresholds over time once they were, I guess, restored or improved, or yeah, I guess after, if management kind of measures were put in place, maybe kind of comparing after that. But yeah, I haven't really gone into those aspects of it too much.
What key message would you like policymakers to take away from this work?
That's a good question. I think related to the second chapter more specifically is that there is this approach for estimating thresholds using these kind of reference conditions, and that we can kind of look more at the uncertainty of these thresholds and then also predict thresholds and perhaps scale them up more to kind of more consistently look across different regions. I would say that there is there is this framework in place, and I'd be really interested in applying it to, as the other question said, other kind of data sets and other indicators to see how they compared. So we can estimate thresholds.
There's a question sort of related to that. So where do you hope this research could be most impactful, such as perhaps being used to assess conditions of MPAs?
Oh, interesting. I think maybe to kind of bring it back again to this ICES work that I'm involved with, that I hope that this will be used, the kind of framework will actually be put in place to estimate thresholds and to assess systems based on these thresholds across different indicators and even perhaps across different methods. But I think I'd be really interested to actually see it being applied. And I hope that I've kind of set out the framework. Chapter 2 is under review at the moment, but I hope I've kind of set out the framework in a way that it kind of can be used by others or by me with other indicators and other data to actually kind of run some of these assessments.
Thank you. How easily do you think this approach would scale to national or international scales?
I think that it would depend on, again, perhaps the legislation and what kind of assessment units it uses. But I think you could effectively scale up, like what I showed with the kind of the benthic relationship with kind of depth and longevity of species. You could effectively scale that up to different assessment units if you had those general relationships. Obviously with sediment type, it would be great as well to kind of have broader or much more specific thresholds depending on what the kind of legislation assessment units are, I guess. But you could effectively easily do that with those general relationships.
Brilliant. Surprisingly, we managed to get through all the questions with extra time, which I didn't expect to happen.
Talking so quickly.
Thank you so much, Lorna, for giving such a great talk, and thank you very much everyone for attending. This will be our last science talk of the year, but we've got lots more planned for next year. So yeah, that's it, and have a lovely rest of the day. Thank you.
Thanks for having me.